I slightly changed up the title a little just because I have a little addition at the end of the talk. And I just really wanted to thank the organizers here for the opportunity to speak to you today about my labs and actually a large group of labs work. And just want to here are my disclosures. And I just want to acknowledge, I know usually it's at the end, but this has really been a very large group effort from multiple groups, including groups here at Columbia with David Ho's group. And so I just wanted to acknowledge that up front, and we'll acknowledge that as I go through. Not going to go too much into obviously what COVID-19 is caused by or the virus itself. I just wanted to start off here just because, I gave a talk about maybe about 2.5-3 weeks ago. And it's amazing how we're really kind of in this logarithmic phase here where this morning we have 64 million cases, and this is just data from 3 weeks ago, and 1.2 million deaths 3 weeks ago, now 1.48 million. And obviously the United States, represented here in North America, has been the most affected. And, initially what inspired us to do do the work I'm going to talk about was, initially there was, these prominent respiratory presentations, but we noted as well in studies by our group and others that there were a variety of different manifestations of COVID-19. This is just reflected even in a short series that we put together at NYP Cornell, which showed that upwards of about 16% of patients presented initially with nausea and vomiting, and about upwards of a quarter of patients presenting with diarrhea, which kind of suggested that many organ systems were being impacted by SARS-CoV-2. And, at the time, this is just back in March, we can't, obviously there were a lot of very important open questions, some of which were being addressed today, but obviously, why are some patients asymptomatic or others just becoming critically ill or dying? What specific risk factors augment the risk for severe disease? How do we, augment patient risk for severe disease, which maybe is addressed here with prophylaxis? How do we treat COVID-19? And then, maybe this last question, which is what we were interested in, is what organs are targets for SARS-CoV-2 infection? And, the thing is that as everyone here is well aware, the platforms that we use to study this virus largely have been, cancer cell lines. The most prominent one or cell line that's been used is the Vero, the green monkey kidney cell line. It's been cultured almost for 50 years and is, everyone's aware, it's just not human. It's marked by abnormal proliferation, unpolarized cells, dysregulated gene expression, and having aberrant cell responses. And really what has become the basic platform we've used in the community, the ACE2A549 or CACO2 or CALO3, these are all cancer cell lines. And so, this really makes it difficult to study host virus interactions, difficult to identify host factors that are important in SARS-CoV-2 infection. And really what we all want to be working with is with primary human lung tissue, colon, or liver tissue. And the approach that we take is using pluripotent stem cells, in this case, induced pluripotent stem cells, which basically was initially discovered or created by Shinya Yamanaka in 2006 and 2007 in mouse and human respectively, and have gone on to be shown it can be used to generate almost every cell type in the human body, and obviously can be used for a variety of different applications. And we have shown that you can use this platform as we go from pluripotent stem cells in a stage and developmentally appropriate manner. We can differentiate these into a variety of different cell types. Here I'm just showing you hepatocyte-like cells. And we've shown that these march through in this morphologically, phenotypically, and functionally appropriate manner as they become hepatocyte-like cells, and have gone on to show that we can use these as platforms, not just to study development or for tissue engineering applications, but for uses in infectious disease. And we've shown that this can be used for a hepatitis B study, hepatitis C study, and have gone on to show that these can be used as successful models to study a variety of hepatotropic pathogens, such as hepatitis A, B I already mentioned, but BD co-infection, Hep B, Zika, CMV, and plasmodial infection. And so we collectively took this system, pluripotent stem cells, and ourselves, as well as our collaborators, differentiated these into the different germ layers, definitive endoderm, mesoderm, ectoderm, and then a variety of different cell types, including colon organoids, lung organoids, pancreatic endocrine cells, hepatic organoids comprising of hepatocytes and cholangiocytes, endothelial cells, cardiomyocytes, macrophages, microglia, cortical neurons, and dopaminergic neurons. And obviously, we kind of focused in or honed in on all these different cell types, but we can produce structures that actually phenocopy the multiple cell types that are present within these different organs in lung, colon, pancreas, and liver. And I'm just going to hone in on each of these different organ types to kind of show what can be done and what we can find with these types of systems. So first, we wanted to see which of these different cell types were permissive to SARS-CoV-2 infection. We started off using a very generously provided a reagent from David Ho's lab, a SARS-CoV-2 pseudoparticle that expresses the spike protein on a VSV luciferase expressing internal shell. We can then inoculate these different cell types and see which cell types allowed for viral entry. And you can see here, this is now looking at luciferase expression. You can see that colon organoids, lung, pancreatic, endocrine organoids, lung organoids, cardiomyocytes, and dopaminergic neurons were highly permissive to viral entry. Endothelial cells, less so. And we did not see very robust viral entry with macrophages, microglia, and cortical neurons. We confirm this here with staining for the luciferase protein, just demonstrating that what cells are being potentially supportive of viral entry. Now, obviously, that's all nice and good, but we want to do this with the real virus. So we inoculated these different cell types with the SARS-CoV-2 Washington strain virus, and we're able to show that this is looking at subgenomic RNA expression, that a variety of these different platforms were susceptible not just to viral entry, but to support viral replication, as denoted here in cholangiocytes, hepatocytes, islet-like cells, lung, cardiomyocytes, dopaminergic neurons. We did not find robust evidence of replication in cortical neurons, microglia, or macrophages consistent with our viral entry data. So I'm just going to break up into these different groups. So we honed in on the hepatocyte or cholangiocyte models. This is our differentiation that is hepatocyte bias. So the majority of cells that are produced are hepatocytes, as denoted here with albumin. This is looking at the pseudovirus or pseudoparticle assay, just again looking to see if they support viral entry. And indeed, we do see that some of these cells are capable of being supportive permissive to viral entry, and around 30 to 40% of the cells have this capacity. We see similar results if we look at the cholangiocyte bias protocol, where we produce a majority of just cholangiocytes denoted here by KRT19, and you can see that a smaller percentage are permissive but do support viral entry. Now, is this true with the real virus? So we inoculated with the SARS-CoV-2 virus, and we can see that basically a large percentage of cells are actually infected with SARS-CoV-2, as denoted here by SARS-S, and I'm not showing you here, but also by SARS-N staining. On the left here, we can also see production of subgenomic RNA production, and if we look at the percentage of cells that are supportive of viral infection, at least 80% of the cells were infected. If we do the same experiment with the cholangiocyte biased organoids or spheroids, we see somewhat similar results. A slightly lower percentage of the cells are becoming infected, with only about a third of the cells being SARS-S positive. We do see robust production of the subgenomic RNA. So if we then lyse the cells and then perform transcriptomic, RNA-seq and then transcriptomic analysis, and then align the genomes, the viral genome against, the transcripts that we see against the viral genome, you can see here that we get representation across the whole genome, and this bias here is very consistent with what we see in vivo as well in vitro. So this is consistent, we feel, with robust infection. And if we look at actually the cellular response to SARS-CoV-2 infection, yes, on PCA we see it's quite different, but that's not so surprising. But what we do see is that there's a robust upregulation of a variety of chemokines basically in the hepatocyte system, as well as in the cholangiocyte bias system. And we actually see similar pathway enrichment with enrichment and basically IL-17 signaling, chemokine signaling, cytokine receptor interactions. And this is consistent across the different infection models. Well, I know I started with the hepatocyte because obviously I normally work on the liver, and that's obviously where my bias forms. And we did see, and I didn't go deeply into the clinical data, but and maybe I should have pointed it out more pointedly, but there are abnormal liver enzymes suggesting hepatic injury, which is why we looked at the liver in the hepatic system. But obviously the target organ that is of most concern is the lung. We can generate lung organoids robustly from pluripotent stem cells and can generate both lung progenitors and epithelial cells. If we perform single-cell RNA-seq at the endpoint of this differentiation process, we can see that a variety of different cell types are produced in the context of these differentiations. And yet specific cell types express ACE2, TMPRSS2, furin, not shown here, also NRP1. And basically there is production of AT1 and AT2 cells. Now, if we then inoculate this again with the SARS-CoV-2 pseudoparticle, we see that yes, indeed, these cells not only express ACE2, but they are susceptible to viral entry here, as represented by the expression of luciferase by the SARS pseudoparticle, as well as by luciferase activity within these infections or inoculations. So we then did the same experiment now where we inoculated these cultures with SARS-CoV-2, and indeed we see production or presence of SARS-S staining here. I know this is hard to see because these basically form balls. I'll show you some additional staining in the next slide. But basically, we also see here on the RNA level production of the subgenomic RNA. And then if we do lysis and then perform RNA-seq and transcriptomic analysis, we do then again see alignment across the whole viral genome. If we look at on a volcano plot here, we see again the same phenomena where we see upregulation of a lot of chemokines without a very robust interferon response. And if we look at, again, the pathway enrichment, we again see this IL-17 signaling pathway, TNF, lumen, and other pathways that we saw previously. Now you can tell me maybe this is just some artifact of our stem cell organoid type model platform, and I think that would be fair. And so we wanted to compare this to autopsy material. And so in collaboration with Alan Borchek, we created a very large autopsy series encompassing over 53 patients where a variety of different organs were isolated at the time of autopsy. And if we compare just 7 of those COVID-19 lungs compared to the healthy, we see similar enrichment here for chemokines. And again, when we look at pathway enrichment, we see this IL-17 signaling pathway present. Yet again, similar to what we saw in our lung organoids. So my collaborator, Xuming Chen, who is very good with doing large chemical screens, performed the chemical screen on these lung organoids, looking to see which ones would potentially impact SARS-CoV-2 entry and infection. We identified 3 that had very good, robust inhibition of both viral entry and infection, both imatinib, mycophenolic acid, and quinacrine. And what I want to point out is that these EC50s are levels that are actually able to be obtained in blood, so they're clinically relevant. Just to show you how this looks, this is in the lung system. If we pre-treat the system, which is the way the screen was performed, we do indeed see that quinacrine, mycophenolic acid, and imatinib significantly inhibit viral infection. But obviously that's not the way things would happen in the real world. So we pre-infect the cells and then come back with these drugs 48 hours later. We do see a similar inhibition of viral replication and over time the percentage of cells that are infected. Now, I know this is hard because these are just, single layer balls because this is a confocal slice. The colon organoids are much more solid. So the same experiment here is done with the colon organoids. And you can see that these three drugs also similarly inhibit viral infection, whether they're treated before or after inoculation with SARS-CoV-2. And in the lung organoids or in vera or in colon organoids, the drug concentrations, particularly for imatinib, are in a range that are obtained clinically. And so basically this drug is right now being evaluated in clinical trials, both in Europe and in the United States. Okay, so I'm gonna now move on to a kind of a different question. And so one of the things that we were really surprised by was that one of the most permissive cell types that we actually saw, at least on the SARS-CoV-2 viral entry assays with the pseudoparticle or with SARS-CoV-2, were actually pancreatic islets. And so pancreatic islets or endocrine cells can be generated from IPS. You can see this on the single cell data. And I already showed you earlier, yes, they express ACE2, and they support viral entry with a pseudoparticle. That was kind of that big agglomerated figure earlier. And if you look here, both the alpha cells denoted here with glucagon or beta cells with insulin both support viral entry and similarly support viral infection with SARS-CoV-2. This is looking at insulin positive cells, SARS S staining, which co-stains both with insulin and glucagon, suggesting that both alpha and beta cells can support viral infection. If we look actually across here, across the whole, if we perform RNA-seq and then do that alignment against the whole viral genome, you can again see that we do get robust alignment across the whole viral genome, showing that we get replication and representation of this. And again, if we do the volcano plots on the host genes that are altered in infection, we see I think I'm missing a slide here, I apologize. What you would see here actually is that the infection leads to a massive upregulation of chemokines as well as alteration in insulin and resistance. And we actually see the same thing here from the some autopsy samples here as well. This is the autopsy here. I apologize, it just got mixed up here, but this is what we would see on the pathway enrichment for the pancreatic endocrine cells. So, the one, again, criticism you can come out with would be, well, maybe this is due to the fact that these are stem cell derived. And I mean, that would be fair. So we actually obtained primary human islets. So these are primary human adult islets. You can see here the expression of insulin glucagon as well as ACE2 across the variety of different cell types present within the islet. Similarly, if we perform a single cell, we can see that there's there's furin expression, actually very low TMPRSS2, little TMPRSS4, and some cathepsin L across the different cell types present within the islet. And if we do infection with SARS-CoV-2, we actually see that essentially the alpha and beta cells both support viral infection as well as some of the other cell types, which I'm not going to talk about today. If we zoom in here at the islets, we can see that the beta cells here represented by insulin or alpha cells represented by glucagon are being infected, and there are cells here that express SARS-N as well as a few other cell types, which we can talk about offline. And we see, again, looking at the individual cell types, or globally, we can see in the presence of SARS-CoV-2, we see production of a variety of chemokines. And this is on the RNA level. We actually then looked at ELISA and confirmed this as well. And if we then look at the pathway enrichment here, we see a variety of upregulated pathways. And now, because it's a little later in the year, that the coronavirus pathogenesis pathway is actually now included in this pathway enrichment. So one of the surprising findings we had here is that we know the cells are under a lot of stress. That's recognized here with the pathway enrichment. But what we actually found was, this is now looking specifically at the beta cells, so insulin producing cells. They don't normally express glucagon, but on the RNA level, we actually saw a significant increase of glucagon transcripts as well as the whole alpha cell transcriptional network. And you know, we thought this was a little surprising, but if we actually then did staining for glucagon, we actually saw that some of the beta cells, so the insulin alone expressing cells, were co-producing you know, essentially both insulin and glucagon. You can see this here in some of the yellow cells. You can see you don't really see this in the mock infected cells. And if we quantify this, we can see that there's a large percentage of co-expressing glucagon and insulin producing cells in what should just be beta cells. The other thing we see is a diminishment in insulin expression on a per cell basis as compared to the mock infected. And so, you know, this, again, one could argue maybe this is just an artifact of an in vitro model system. Now, we've seen this with the stem cell derived system. We see this in the primary human islet system. Well, does this actually happen in people? And so this is now looking at the autopsy material from patients. You can see here, you know, insulin and glucagon staining, and we do indeed see some SARS and staining in the islets. Not saying it's a super common event, but we do see that. And we also see some glucagon and insulin co-expressing cells. And so if we quantify this, you know, we essentially first see diminishment in insulin intensity, and we see a small percentage, I'm not saying it's all, but it's upwards of about a third of the insulin positive cells co-express glucagon, similar to what we actually saw with the primary human adult islets, as well as the iPS-derived islets. So I'm just going to skip that, just, but we can talk about that if anyone has any questions later. But so we actually then did experiments where we actually controlled the timing, because obviously, you know, autopsy, you know, the sickest of the sick, you know, obviously we don't control where in the illness people die from their infection. So this is an example of an experiment we've done in hamsters, where we infect, you know, basically hamsters with SARS-CoV-2, and then we can sack these animals or euthanize the animals as we go along. And we actually see, again, infection in the islets, and we see some co-expression, we do see some co-expressing cells here. And so right now, this is a work in progress. So we're still investigating this as we speak. And, you know, basically stay tuned on that. So I'm just going to rush through the last organ system because I really do want to get to the autopsy series, just a brief discussion on that. So we did look at cardiomyocytes, and, you know, I showed you this data that cardiomyocytes are robustly permissive to this virus. And, you know, we were quite surprised by this, but it's actually, it seems to be the most permissive cell type to SARS-CoV-2, with absurdly high viral loads produced after infection. And we see this represented here just across on the transcriptome alignment after RNA-seq. We see that a large percentage of the cells are infected. They're robustly infected. Again, production of chemokines and a variety of different signaling. And we see this is IPS.
2-minute warning or so, Rob.
Okay, you know, I'm just going to skip over this. Cardiomyocytes are permissive, and we can phenocopy that and see that in autopsy material. So I just want to talk about the last thing here, just because it's been a really powerful tool, and it's something we've been really excited about. So as many of you are aware, you know, one of the big challenges when one does immunofluorescence or immunohistochemistry is spectral overlap. So it really limits how many markers you can work with at the same time. And so imaging mass cytometry liberates you from this problem, because you essentially link antibodies to heavy metals, and similar to Cytoph, you can essentially work with upwards of 38 to 40 different markers. And the nice thing about imaging mass cytometry is you get the spatial information as well. So in this autopsy series, we actually looked at 4 healthy patients with who had died from influenza or sepsis or bacterial pneumonia, and compared them to patients who died early or late from COVID. And basically what we can show is we developed completely different new algorithms to try to structurally analyze these lungs. And not unsurprisingly, COVID seems to stick out as compared to even ARDS or other disease entities as to the amount of fibrosis that is present, as well as just the overall architecture, which is completely altered in this disease process. I mean, and this is just reflected here, where you can see even in the lowest fibrotic states barely compare to the highest fibrotic states that we see in flu-induced ARDS or even in ARDS. So that's the first thing I just wanted to point out. I'll go 1 minute over, I apologize. Basically, there's 664,000 cells that we analyze across all these different disease states, and we can actually bin these cells based on their marker expression. These are kind of the macro bins just of cell types. We actually identified 63 different cell types using the different bins, essentially, of markers that we had. But if we break them down less concretely just into these different bins, you can kind of then see first which markers are expressed across these different cell types in these different disease states, and also in which disease states these different cell types are enriched. In this case, like if you just pick on macrophages, they're enriched in late COVID disease as compared to all the other disease states. And you can do this for each of these individual bins. We can then get a little more granular here, looking at the different bins here, like macrophages, neutrophils, specific types of T cells, or even go a little more deeply looking at the different types of macrophages. And you can see here that interstitial macrophages, which are the non-tissue residents that recruited, are significantly increased in late COVID as compared to all the other different disease states. These are just giving you an idea of all the different types of cells that are represented here and how they can be potentially represented or enriched in the individual disease states, such as in COVID late. I'm just picking one here. Essentially epithelial cells that are infected with COVID, not surprising that's in the COVID disease state. The last, three things I'll say on this, we can then look at how the infected cells are different between infected and uninfected cells in patients who have COVID. And what we found here is that there's an enrichment of a variety of different markers, but specifically what we note is that a significant number of the infected or SARS-CoV-2 positive cells are marked by active complement, as well as a variety of other immune or surrounded by immune cells. And so the nice thing that this technology can afford us is we can actually look to see which different cell types are near other cell types, their neighborhoods, as well as their adjacent cells, and then look to see how that geometry or organization of cells is altered or changed in the context of these different disease states. Now, I know this is pretty, but it's hard to then interpret, but what you can see here is that we can actually then see which different cellular interactions are enriched in COVID late versus early, or COVID versus healthy, or COVID versus flu, to try to get an idea of how this disease entity is unique or different or similar to what we've learned, what we know of from these other disease states. And this is just magnified here. I'll just leave it there. Can talk about this offline. What I think I've shown you today is that iPS-derived organoids express ACE2 and TMPRSS2. They support viral entry. They support viral infection. We can enable us to do a variety of drug screens. Also allows us to better understand the life cycle, which cells are involved. And we're right now trying to use this to understand which host factors are involved in this process, the impact of viral polymorphisms on infections. We've developed now multicellular organoids with immune components, and we're now also comparing this deeply to the autopsy series that we've developed to better understand COVID disease. And just want to acknowledge the many groups that have participated in this, work very closely with Shubing Chen, David Ho's lab, Benjamin Tenover's group, the autopsy series with Alan Borchek, Stephen Salvatore, and Olivier Elemento's group, and a lot of the drug work's been done with Luke Hantley's group. With that, I'll gladly take any questions.
All right, Rob, thank you very much. Very cool. So, it seems clear we're now appreciating that the virus is replicating in all kinds of organs. And I guess one has led to the idea that the damage is very direct, that maybe we're just, having direct viral cytopathology in those tissues. But I wonder to what extent we can say how much of the damage is immune-based in response to the virus or direct viral effects.
So I didn't get into that data, but what we see, like for example, in the cardiomyocyte model, is that the cardiomyocytes get injured from SARS-CoV-2. They upregulate ROS. A bunch of cells will upregulate, apoptotic pathways, so cells are lost. But the real injury we see is when in the immune organoid models. So what we find is that if we incorporate macrophages or other immune subtypes into the model, we see much less actually virus in the system, that there is greater control of viral replication or less viral replication, I should say. But what we actually see is that those cells are killing the infected cardiomyocytes. We see that in the lung model, we see it in the cardiomyocyte model, and we've seen it in the liver model. So I think that, I think that some of the injury is directly coming from the virus, and that might portend problems later on, like sequela maybe down the line. But then there's also this immune component, which I think is causing the initial direct injury during the acute illness itself. And so I think there's two questions there. One, what is going to be the long-term sequela of having been infected, even if you had a mild infection? Because our concerns from like the pancreas data would suggest that maybe these patients would be at risk for developing type 2 diabetes. It's just a possibility down the line with loss of beta cells. And so I think there are a lot of open questions.
So we had one related from Lily Yamasaki who's asking about what about cardiomyocytes and affected children.
I mean, I think it's a great question, and I think it's one of the concerns that we have. I mean, we've been able to verify in our autopsy series, at least. Obviously, we don't have any kids in that autopsy series, and we don't have a lot of biopsy material, in the acute illness. But I can say that we have a biopsy from a very young adult. The patient was 19, and another patient that was 33, and it's chock full of virus. They died in the acute phase. Sorry, one of them died in the acute phase of the illness. The other one, they died from myocarditis, which we believe is probably from SARS-CoV-2. So I think the question asks what the long-term sequelae are going to be, and I don't know that yet.